I'm Lizzie, and today I'm going to be discussing how zebrafish are being used by scientists to help understand the mechanisms behind fetal alcohol syndrome disorder. Fetal alcohol syndrome disorder, or FAS, refers to a range of conditions that result from prenatal alcohol exposure. At the most severe end is fetal alcohol syndrome, or FAS, which is characterised by distinctive facial abnormalities, growth restrictions, and central nervous system damage. These early structural problems translate into lifelong difficulties with learning, memory, behaviour, and social functioning. FAS is therefore considered one of the leading preventable causes of neurodevelopmental disabilities worldwide. How does alcohol affect the fetus? When alcohol is consumed during pregnancy, it easily crosses the placenta, and because the fetal liver can't metabolize it effectively, the fetus experiences prolonged exposure. This has multiple damaging consequences, as ethanol causes direct cellular toxicity, triggers neuronal cell death, generates oxidative stress, and disrupts key developmental signaling pathways. It also alters gene expression, which affects how organs and brain structures form. Symptoms of FAS are diverse but usually fall into four categories. First was facial features such as smooth philtrum, thin upper lip, and small eye openings. Second are growth deficits including lower birth weight, smaller stature, and reduced head circumference. Third are neurological impairments including intellectual disability, learning difficulties, and attention deficits. And finally, there are behavioural and social problems such as hyperactivity, anxiety, depression, and poor judgment. Studying FAS directly in humans is extremely challenging. There are serious ethical barriers. It is not permissible to experimentally expose pregnant individuals to alcohol. Self-reported alcohol consumption data is often unreliable. And finally, alcohol's effects are difficult to isolate from other maternal factors such as nutrition and stress. Why zebrafish? Zebrafish overcome many of these barriers and are excellent models for studying FAS. They share about 70% of their genes with humans, including most known disease genes. Their embryos are transparent, which lets scientists directly observe developmental processes in real time. They also develop very rapidly. Major organ systems form in less than 2 days, making experiments efficient. Zebrafish metabolize ethanol using similar enzymes as humans, ensuring biological relevance. And finally, they're cost-effective, producing large numbers of embryos in a short time. This study validated the zebrafish as a reliable model for studying fetal alcohol syndrome disorder. Zebrafish embryos were exposed to a 0.5% ethanol concentration during a critical developmental window of 2 to 24 hours post-fertilization. The research analysed 3 key areas to show that ethanol exposure led to physical, behavioural, and genetic changes similar to those seen in adults with FAS. As you can see, the fish had reduced eye and skull size, enlarged heart chambers, impaired sensory motor behaviour, and inflammatory gene dysregulation. The experiment exposed zebrafish to a 10% ethanol solution for just 1 hour at different developmental stages. The results were striking. Early exposure caused more than 88% mortality. Survivors developed severe malformations, particularly during the PRIM16 stage, with nearly 85% showing deficits. Sensitive organs included the eyes, ears, and yolk sac. This shows that vulnerability depends heavily on the timing of exposure. Another study examined embryos exposed to varying ethanol concentrations. Using fluorescent imaging, researchers tracked cell movement during gastrulation, a critical stage in forming the body axis. The results showed a clear dose-dependent effect. Higher ethanol concentrations cause more severe abnormalities, higher mortality, and delays in cell movements required for proper development. This provided direct evidence that alcohol interferes with fundamental cellular layout. At a molecular level, ethanol disrupts the expression of over 500 genes, including a significant reduction in the transcription factor SOX2, which regulates essential pathways for development. Ethanol exposure caused defective epiboly, where cells fail to spread normally over the yolk. Remarkably, when SOX2 mRNA was reintroduced, many of the defects were partially reversed, suggesting potential avenues for therapeutic interventions. Altogether, these studies reveal that ethanol first suppresses gene expression during gastrulation, then disrupts critical cell movements in gastrulation and neurulation. This leads to craniofacial malformations, reduced neuronal numbers, and disrupted axonal growth. Essentially, ethanol derails the precise developmental program that an embryo depends on. Despite these strengths, zebrafish are not perfect. There are metabolic differences, so alcohol concentrations don't always translate directly to humans. Zebrafish also lack a placenta, meaning maternal-fetal interactions are not fully replicated. Their behaviour is simpler than human behaviour, which limits studies of higher-order social and cognitive effects. And finally, FASD in humans is complex and influenced by many environmental factors, making it difficult to model fully in fish.